session we are just continuing with our previous classes diagram. But before going to other diagrams, I just recap all the things what we already studied in our previous classes. So in our previous class we saw that in development of dicot embryo, the first stage is your zygote. Nextly, it gives a mitotic division, and from the mitotic division two cells are formed, and the cell which is towards your chalaza end form your terminal cell, and the cell which is towards your microvillar end form your basal cell. Nextly, the basal cell forms your transverse division, and a stock of cells are produced. That means it looks like a stock of coin, and 7 to 8 number of cells are formed, which is always known as your suspensor cell and a hostile cell also. Nextly, the terminal cell is divided, and it divides in all plane, but the basal cell divides in transversely. So as it started divided in all planes, so a mass of cells are produced, and it looks like a ball. But in primitive cell stage, 4 cells are formed. So the structure is otherwise known as a proembryo. As it forms 4 cells, so it is quadrant. So it is otherwise known as a proembryonal quadrant. And nextly, the 4 cells which is formed over here, they again change to form 8 cells, and the cells is otherwise known as a proembryonal octant. Here is your 4 cells, so quadrant, 8 cells, so octant. Nextly, the cell which is attached to your proembryo, that means the first cell of your suspensor, going to change to form your hypophysis, and the other parts remains as it is. That means your suspensor cell is as it is, and your posterior cell also as it is. Nextly, the host hypophysis, that means the cell which is attached to your lower part of your proembryo, is again changed to form 8 cells. That means 8 cells are formed over here, and the lower 4 cells, that means these 4 cells which is towards your microvillar end, also you may call it as the cell which is attached to your suspensor cell, is form your root cap and root tip, and the cell which is just attached to your lower part of your proembryo is already formed your root cortex. That means the total structure changes to form your radicle. That means this total structure form your root part. And nextly, the changes takes place in your proembryonal state. That means a periclinal division takes place in your proembryo, and as a periclinal division takes place, so every single cell again divide to produce a layer over it, and the sideways layer is already known as your dermatogen, and the dermatogen again produce your epidermis. That means these are the fate of these cells. That means I also told you that the structure is not look like that. We just assuming these structures and how the changes are taking place. These are the fate of these cells. So we already covered the proembryo. Nextly, the changes which is occur in your proembryo. The changes already occur in your hypophysis. Nextly, the proembryo. So the lower 4 cells, that means these 4 cells, 1, 2, 3 and 4. These 4 cells change to form your hypocotyl. That means your lower part of your proembryo changes to form your hypocotyl, and the upper 4 cells of your hypocotyl form your plumule and occluder. That means 2 changes are expressed. So what happens here? The 4 cells towards your upper part of your proembryo changes your plumule and cotyledon, and the lower 4 cells forms your hypocotyl. And this structure is otherwise known as your globular embryo. So the structure is your globular, and the globular structure is already formed. So we just continue with the same diagram. So consider that the proembryo is as it is. So we just continue with the same diagram. So here is your proembryo, and the divisions are takes place. What happens next? The hypophysis is also formed. These are the cells, and the periclinial division is also set takes place at the and the dermatogen is formed, the outer layer, which is or as it changes to form your epidermis. Nextly, changes takes place in your hypophysis. So there is 8 cells in hypophysis. We just draw all the 8 cells first. So these are the 8 cells. 1, 2, 3, 4, 5, 6, 7, 8. So here is the 8 cells of the hypophysis. So what we studied so far? We studied that the lower cells which are attached to your suspensor, they are changes to form your root cap and root tip. We all know that. So the cells which is over here, that means your root cap and root tip cells, they are going to divide rapidly and they bend little bit. As they are roots, they form a root, so they bend little bit and forms a structure like that. So here is the mass of cells are formed. The cell division takes place and the mass of cells are formed over here. So you just draw the mass of cells first. So here the mass of cells which forms your root tip or root cap. Nextly, the upper 4 cells which is attached to your proembryo, that means the lower part of your proembryo again change to form your root cortex. That means the 4 cells which is attached to the lower part of the proembryo. That means these 4 cells. So we just erase it, and they are also going to divide and form the root cortex. We just change these 4 cells and elongate it. Nextly, these 4 cells, they form your root cortex. So here are the cells which are again divided to form the root cortex. So here what happens? The root part is already developed. That means the cell division is takes place and the elongation is also takes place. So root is started to develop. Nextly, that means the radical part is developed. Root is not developed, the radical part is developed. Nextly, the lower part of the proembryo, that means the lower half structure, that means these parts, these structures. The lower half structure is again going to divide and this is take a bulging structure, that means which forms your cotyledon. So we just erase these cells. So in the total structure, that means in the total structure of 4 proembryo, 3 structures are going to divide. One is your hypocotyle, another one is your plenum and cotyledon. So the lower half structure forms your hypocotyle. So same cell division is takes place and the hypocotyle is formed. So here the cell division and a mass of cell is going to form and the lower half is filled with the cells. So it is the hypocotyle. We just not level it, later we just level it. So here is the hypocotyle. Here is the root cortex and root cap and root tip. So it is the radical part, and we just started drawing the changes of your proembryo. Nextly, the other half structure of your proembryo, that means the upper half surface of your proembryo again started to form your cotyledon. That means we just studying about your dicotyledon. So 2 cotyledons are going to form and they just start to divide. So what happens here? There is 2 bulging structures are going to form. So they are just started dividing rapidly and forms two bulging structures. So just erase these cells first. And so what happens here? There is the bulging structures are formed. So if we look the diagram, somehow it looks like a heart. So here is the cotyledons are formed, and I told you plumule is also formed. So here is the plumule. So the structure is look like a heart. So this shape is always known as a heart-shaped embryo. So here is the formation of your heart-shaped embryo formation takes place. First globular embryo takes place. Nextly globular to heart-shaped embryo takes place. Nextly, as we are studying that a dicote embryo and these all structures are found in a teeny embryo cell. That means in a teeny embryo cell, these all structures are going to develop. So there is no enough space. That means there is a scarcity of space takes place. So the bending of the cotyledons are takes place. So I just bend the cotyledon. When it started dividing rapidly, there is not enough space, so it bent down. So there is your first cotyledon. I just leveling one cotyledon. There is also another cotyledon. As we are studying dicot, so two cotyledons are. So here the first cotyledon, it started to bend like that. I just change it. There is one bending and there is your another bending. So your first cotyledon is bent. Nextly, your second cotyledon. It is also bent like that. You just overlap a little bit, just manage it. So, if you look these two structures, that means the cotyledon is again bent and this structure is look like a horseshoe. So this structure is known as your horseshoe shaped embryo. Why this horseshoe shaped embryo form? Because there is not enough space in your embryo cell. So as the rapid division takes place, so it just bend down and the middle one is a plumule. So this shape is known as your horseshoe shape. So if there is a question, if asked to you, the sequence of your development of embryo. The first sequence is your globular. The second sequence your heart-shaped, that means from this to this is your heart-shaped embryo. Next is the total structure, which is your horseshoe-shaped embryo. So it is the sequence. Nextly, how this formation takes place. We just rapidly look after a cotyledon and how a dicotyledon and how the structures are formed, how the embryonal axis is formed and all this. So consider that a cotyledon over here. So these are the cotyledons. As we are studying about your dicot, so two cotyledons are there. So here is your I just show you how the seed is developed. That means how the embryo is going to develop to form a plant. We just studying about the structures which are formed. So the lower part is that means your lower tip. This is your embryonal axis, and this the lower tip is already known as your radicle, and the upper tip is already known as your plumule. I told you that here is your plumule, and the lower part form your radicle. So radicle is formed, plumule is formed, and the cotyledon is formed. Nextly, the point where the two cotyledons are attached. That means the point where the two cotyledons are attached are otherwise known as your cotyledonary node. So here is your cotyledonary node. Nextly, the place, that means the place here, that means in between your radicle and your cotyledonary node, that means where the attachment of your cotyledon, that means the place left here, that means this place. Here is your radical part, here is your cotyledonary node. So this place is otherwise known as your hypocotyl. So here is your hypocotyl. Nextly, the structure which left between your plumule and your cotyledon, that means the upper surface, is always known as your epicotyl. In morphology, you all know that how the seedling is takes place, how the seed is developed, and this is the structure. You just assume when all the things are going on, you just assume in your mind that how the structure is formed, how the cells are going to divide, and the structure. For the division only, we just draw these types of diagrams. These are only the structure, that is the fate of the cells, and this is the actual structure where it is formed actually. So if we take all these things, this total structure, that means this total axis is already known as your embryonal axis. And this embryonal axis contains your plumule, radicle, epicotyl, hypocotyl, and two cotyledons, and a single cotyledonary node in which the cotyledons are attached. And if we take a common example, we just take an example of your P. So in P, you know that if we draw an example of your P pod, I just roughly draw an example of your P pod. So here the P's are attached, and if we take one P, that means one P seed, we just open it, then two seeds are developed. That means there is a half circle-like structure. Half circle-like structures, and these circles are attached in a white tiny part. This white tiny part is the total embryonal axis, and the half structures which are developed, you may take it as your example of your bean seed, you may take as your pea seed, or you may take as your gram seed, whatever it may be. The white structures are known as your embryonal axis, and these two structures, that means the two half of the seeds, half part of your seeds, in pea seeds the two half seeds, they are always known as your cotyledons. So I hope these all structures and all the diagrammatic representation is helpful to all of you. So in our next class we are going to study about the monoclone. That means we already studied our development of a dicote embryo. In our next class we just continue with your monoclone, development of the monoclone. Still, stay tuned. Thank you.